Hello, my name is Mr. Chipman, and I teach biology and AP Biology at Murray High School in Murray, Kentucky. This is 5.4 Non-Mendelian Genetics. We talked about Mendelian genetics in the last video, and so in this particular video, we are not talking about Mendelian genetics. You probably figured that out. So Mendel came up with these principles, right? Dominance and segregation, independent assortment. And there's very clear rules, very clear expectations. I think it's a better way of explaining it. And so when the results do not meet our expectations, we typically lump it into this concept of non-Mendelian genetics. Here's an example. And so here's an example of non-Mendelian genetics. You have a trait, which is color of rabbit fur. And it's not just a simple dominant recessive, black and white, right? You have some intermediates. And so instead of just dominant recessive, you have most dominant, most recessive. You have second place, and you have third place, which is chinchilla. Or maybe chinchilla second place. I guess it just depends on how you look at it. But you get the idea. So Mendel didn't account for this, right? He didn't account for this idea that one gene can have more than two alleles. And so when we have something like this, something that doesn't quite fall into that normal Mendelian paradigm, we call it non-Mendelian genetics. The first of those is everyone's favorite, linked genes. Linked genes is not everyone's favorite. Linked genes is sort of a difficult, well, it's an easy concept, but some of the coming out information of that is going to be more difficult. So linked genes, easy concept. These are genes that are found on the same chromosome, right? And that's very simple concept. In this particular case over here, A and B are not linked because they're not found on the same chromosome. There's no chance that A and B are going to cross over. Big A and little a might cross over, but big A and big B are not going to cross over, right? That idea of crossing over is another idea that we call that is recombination. Over here, A and B are on the same chromosome, so they're linked together, right? And there is a chance that big B would not be inherited with big A here because there could be crossing over, and there could be a chance that big A and little b would be inherited together because of that crossing over event. And so that is one of the concepts here that you need to be familiar with is the idea of recombination frequency. Recombination frequency is the frequency at which those genes will cross over. And that frequency typically has to do with the distance that those genes are apart from one another, right? That distance is usually looked at in a percentage, and then that percentage is then transferred to something called a map unit. And so if it's a 1% recombination frequency, meaning that there's only 1% chance that A and B would not be inherited together, then that means it's one map unit apart, right? It's very short distance apart from one another. Whereas if it's really far apart, if 60% of the time they cross over, then there's a 60 map unit distance between those two things, right? And so a lot of times the way this is going to sort of rear its head is you're going to have a problem, and you're going to see you're going to have like A and B, and you're going to have expected ratios between those two things, right? I expect to see 9331, for instance, like we talked about in the last video. Well, what if you don't see 9331, right? Then it's not Mendelian, and there's a chance that it could be this concept of linked genes, right? A good rule of thumb is if the recombination frequency is less than 50%, meaning that if two genes are combined together less than 50% of the time, then those genes are likely linked together, right? And so here's an example of this. Genes close together on the same chromosome, these are linked together. Big A and big B are inherited together 50 or 48% of the time, right? And they recombine at 2% of the time. This is less than 50%, right? And so then you would say that A and B are linked. If it's more than 50% or 50%, then you would say that those genes are not linked, which is what you'd expect to see here, right? That's just a general rule of thumb, but when it doesn't meet those expectations, linked genes is a possible culprit. What are some other culprits? Well, codominance is a possible culprit. Codominance is the idea that you have more than one dominant allele, and both alleles express themselves, all right? And so in this particular picture, you have red is dominant, white is dominant. even though they used a lowercase letter. I know it's confusing, but they're both dominant. And so if they mix together, if you have red dominant and white dominant come together in one individual, you have a red-white plant. Both red and white are fully expressing themselves. Co-dominant, they're sharing dominance, right? It's the way to think about it. Incomplete dominance is where you have more than one dominant allele. Big idea. That's the big idea, more than one dominant allele. But in this case, they're not completely dominating, right? And so if red and white come together, then you don't have completely red and you don't have completely white. Instead, you have a blend of those two things. You have pink. And so incomplete dominance, co-dominance, very easy to get those mixed up. Just remember, co is sharing dominance. Incomplete is not completely dominant. Exactly what the word says. Don't get those two things mixed up. Sex-linked inheritance, we talked a little bit in the last video, so just kind of go over it again. Sex-linked inheritance is when a gene is located on the X chromosome. Could be on the Y chromosome as well. Not likely to see that, right? And if it's located on the X chromosome, it creates some interesting things because males only have one X chromosome, and they have a Y chromosome as well. The Y chromosome just has genes on it that determine maleness, for lack of a better term. And so here, the father has a dominant allele. He can only either have a dominant or recessive allele. He can't have both. So if he has that dominant allele, all of his daughters are going to have a dominant allele. There's no way that a daughter can have a recessive condition if dad is dominant for that condition. If mom is dominant for it but carries that recessive allele, then there's a chance that their sons would have that because sons always get their X chromosome from their mom. And so if you know those simple rules, dad always gives one X to their daughter, and the daughters are going to have that X if it's dominant or recessive. And sons only get their X from mom. There's no way that dad can give his X to mom or give his X to his sons, because if he gives his X to his sons are daughters. You get the idea. And this is just showing you, I think it's important to note that when you do a sex-linked problem, not only are you dealing with the trait at hand, whether it's hemophilia is a common one, colorblindness is another common one. Those are both recessive. Marfan syndrome is a dominant sex-linked disorder. But you're also inheriting X and Y chromosomes. So it's a kind of dihybrid cross, but you can really set it up simply by doing a Punnett square like this, like you see here, showing the inheritance of the X and Y chromosomes and the H gene here for hemophilia. Pleiotropy. Pleiotropy is where you have one gene that can affect multiple phenotypes. There's several examples of pleiotropy. I mentioned Marfan syndrome. Marfan syndrome is where you have a X-linked dominant disorder, and in this particular case, Marfan syndrome is going to be one gene, right? But it affects connective tissue, it affects your heart, it affects your eyes, it affects your skeleton. So that one gene is going to affect multiple different phenotypes. This is called pleiotropy. Non-nuclear inheritance. That sounds weird. Nuclear here is not like nuclear power, but it is like nucleus, right? It is like the nucleus of a cell. So where is some non-nuclear DNA? It's found in the mitochondria. And so you can have mitochondrial genes that are inherited. In fact, we all have mitochondrial genes inherited, and they only come from one place. They come from your mother. And so you can detect a non-nuclear inheritance if it's something that is passed only from the mom, right? And that is, so you can kind of see it on this chart here. Mom has it, and so all of the offspring are going to have it through the mother, right? Not the father. Blood types are another thing that you'll see. Probably A and B are co-dominant with one another, so if you have them together, you have type AB. O is recessive, and so if you have AO or BO, then you have type A or type B, right? You can be AA or AO and be type A, right? The only way to be type O is to be OO. And then there's positive negative. Positive negative is this whole other thing. It is not ABO, right? It is tied to those. It's tied to that, right? You can be A negative, but if you're A negative, it's because you have a dominant A allele and you have two recessive alleles for negative. Negative is recessive, dominant is positive. I said it correctly. Moving on. We talked about these pedigrees already, so I'm just going to kind of leave this here for you to look at. But I think the ones that students have most trouble with are the sex-linked ones. And so again, you're just using those simple rules, right? Dad has this dominant disorder, which is blue here, or not disorder, just gene, right? And so the daughters are always going to get that dominant gene, right? Mom may or may not have it, right? Here's this white is represented by heterozygous here. So this daughter is also heterozygous. How do I know? Dad gives her a dominant, mom gives her recessive. This son is either dominant or recessive, right? We don't know. We have to look at his children. Well, this mom is recessive, so she only gives away recessives. And so the sons then are going to be dominant. How's that work? It's because I don't understand these colors. That's what it is. But we can look at this. This is an X-linked recessive. The sons have dominance here and the daughters have, So here we have some example pedigrees. You can kind of look at these and, I encourage you to sort of parse through each one, maybe pause the video and look. We're not going to spend a lot of time on them, but there will be pedigrees on the exam. You will have to be able to sort of look through them and understand what's going on as you do that. All right, so let's look at a practice problem. Intercross between two heterozygous roan cows gives us what roan is. Roan is like a mix between red and white, where red and white are both dominant. Co-dominant was the expected phenotype. And so we know that RW can give away an R or W. So what is the chance that RR and RR is going to be? Well, it's a quarter of the time, right? 1/2 times 1/2 is a quarter. We know that there's a chance that they're also going to be roan, because this one could be little r, big R, and this one could be W. You get the idea. And there's a chance for WW. So we're going to see a quarter, and a half, which is the first choice here. Don't have ABCDs on them. It's a special one. I hope this was helpful. Non-Mendelian genetics, particularly linked genes, encourage you to look at some practice problems with this because it can be a little bit difficult. But again, if you understand that rule about the 50%, you should be just fine. Leave me some comments if you have questions. Thanks for watching.